Welcome to Curiosity, the podcast where science meets wonder. Join us as we explore the fascinating, the innovative, and the unexpected, from the secrets of the cosmos to the marvels of everyday life. Each episode takes you on a journey to uncover the knowledge and stories that fuel our endless quest for understanding. Let your curiosity guide you.
Welcome to the Deep Dive. Today, we're peeling back a layer of reality that is, well, perhaps the most profound and invisible one of all. Forget thinking of yourself as just, a singular organism. You, me, everyone listening right now, we are basically walking, talking superorganism.
It really is a foundational concept, and honestly, we're only just beginning to grasp it. We are living in this intimate, fundamental partnership with trillions of microscopic inhabitants. This whole collective is known as the human microbiome.
Right.
And this community, it's really responsible for some of the most revolutionary discoveries in modern science. It's completely changing how we view health, disease, even identity.
Absolutely. Our sources today really lay out the sheer scale of this internal ecosystem. So our mission here is to understand its profound systemic influence. We're going way beyond just like simple digestion. We're talking about its direct role in immunity, metabolism, and maybe most surprisingly, our emotional and cognitive well-being. And we'll also examine some of the cutting-edge ways scientists are actually learning to precisely manipulate this internal world.
And here's, I think, the single most important nugget you need to grasp right now, just to set the stage. It's the staggering realization that you are genetically more microbial than human. Your health, really in every sense of the word, is defined by this partnership.
That idea, genetically outnumbered, it's just fascinating. Okay, let's unpack this. We need some truly staggering numbers here just to give you, the listener, a real sense of scale. The figures regarding our microscopic residents are, well, astronomical.
Yeah, when research first started kicking off, the common estimate was maybe 10 times more microbial cells than human cells. That was the number thrown around. But more precise recent studies, and this is the data our sources are pointing to, they indicate a revised figure. It's around approximately 38 trillion microbial cells.
38 trillion. And what does that mean in relation to, well, us? Our own.
So that number is roughly equal to, or maybe slightly exceeding, the total number of human cells we possess. It depends a bit on the individual, of course. But this updated figure, it really confirms the physical scale of this superorganism concept. And if you look at the sheer biomass...
Like the actual weight.
Exactly. This collection of microbes, it weighs about 1 to 2 kilograms. That's what, 2 to 4 pounds of living, working organisms functioning inside you right now.
That is some heavy biology we're carrying around. Wow. But I think, for me, the genetic influence is the real mind bender here.
Absolutely. If you compare the genes encoded in your human DNA, our basic blueprint, versus the genes carried by all your microbial partners, the microbial genes outnumber human genes by something like 100 to 1.
100 to 1?
Yeah. That's just an immense pool of untapped genetic potential living inside us.
So if our human genome is kind of like a basic operating system, with the core functions, then the microbial genome is this massive 100 times larger library of apps and programs providing all the capabilities we just don't have ourselves.
That's a perfect analogy. Precisely. This vast library of microbial DNA provides essential metabolic and defensive capabilities that we simply don't possess. We effectively outsource many critical life functions, especially things related to processing complex food structures, synthesizing vital compounds. We outsourced that to our microbial partners eons ago.
Okay, so moving from the numbers to the geography then.
Yeah.
These microbes aren't just like swimming around randomly inside us, right?
Yeah.
They have specific neighborhoods they've colonized. We see this microbial geography at play where different body sites need distinct, specialized communities. It's almost like highly regulated microbial real estate.
That's right. The primary habitat, the place with the highest density and diversity, is unequivocally the gut, specifically the large intestine or the colon. That one area alone houses hundreds of trillions of cells and literally thousands of distinct species, all flourishing in an environment optimized for anaerobic life without oxygen.
But the body has specialized microbiomes pretty much everywhere, doesn't it? Like take the skin, for instance.
Yes, absolutely. The skin hosts these diverse communities that are adapted precisely to the physical environment there. You see unique differences between, say, oily areas like your forehead versus dry areas, or moist skin areas like the folds of your elbow. They're different worlds.
Right.
And in the mouth, you get these complex biofilms forming dental plaque. That's a highly specialized, really structured community designed specifically to stick to tooth surfaces.
And this adaptation can be absolutely critical for maintaining health. Very sensitive areas too.
Exactly. That's why the vaginal microbiome is so often dominated by lactobacillus species. They are crucial. They maintain the necessary acidic conditions by fermenting local sugars into lactic acid. And this regulation of the environment, it keeps potential pathogens from getting a foothold.
What's really fascinating, though, is that the map of microbial life seems to still be expanding. Our sources mention sites we once thought were completely sterile, like the lungs or even the bladder. They're now known to host their own unique communities, maybe smaller and less dense, but they're there.
The old notion of internal sterility is just rapidly crumbling under the weight of new sequencing technology. It really is.
So it raises a powerful question for you listening. If life is basically everywhere inside us, what does that actually mean for me as an individual?
Well, it means that while we all share certain core functions, maybe certain foundational species, your individual microbiome is absolutely unique. It's a signature as personal as a fingerprint. Or maybe, more accurately, like the dynamic composition of a really bustling city.
I like that analogy.
The specific composition, and maybe more importantly, the relative abundance of these thousands of species, varies dramatically from person to person. They're influenced by this complex, lifelong history of exposures. Things like your genetics, your lifelong diet, where you live geographically.
Right.
The exact method you were born by, whether you were breastfed, and every single antibiotic exposure you ever had. All of that combines to create this intensely personal ecosystem.
So the history of your life is literally reflected in your gut microbes.
Indeed it is. It's like a living history book.
Okay, so let's drill down into that largest ecosystem then, the gut. Given the sheer size and density we just talked about, it makes sense that the gut microbiome gets the most scientific attention. It's the command center.
It absolutely serves as the functional nexus, yeah. Its location means it's the first point of contact for all the external nutrients coming in, and it directly interfaces with our largest immune system component, which is lining the gut. This incredible diversity isn't accidental, it's really optimized for influence.
And the critical first job is, obviously, digestive assistance. Our human enzymes are great at breaking down proteins, fats, simple sugars, but they completely fail when it comes to complex carbohydrates and fiber.
That's right. The colon essentially acts as this highly efficient fermentation chamber. When fiber reaches the large intestine, that's when the bacteria really get to work. They're the ones breaking down those complex plant polysaccharides that we simply cannot handle on our own. They turn otherwise indigestible stuff into usable energy for us.
But the impact goes way beyond just calorie extraction, doesn't it? These gut bacteria are far more than just like garbage disposal units or fermentation experts. They're sophisticated metabolite factories. They produce these essential chemical compounds, metabolites, that then enter our bloodstream and affect distant organs. It really signals the systemic reach of the gut.
And here is where we absolutely need to focus for a moment. The power of short-chain fatty acids, or SCFAs. This category includes things like butyrate, propionate, and acetate. They are the primary products when bacteria ferment fiber.
Okay, let's focus on butyrate first, because our sources really seem to highlight its critical importance. How do these SCFAs act as, well, you called it a triple threat?
Well, think of butyrate specifically as the highest octane fuel for your gut lining itself. It serves as the main energy source for those intestinal cells, keeping them healthy, maintaining the integrity of the gut barrier. Without enough butyrate, those cells effectively starve, and the barrier starts to weaken, becomes leaky.
Okay, so that's butyrate feeding the gut wall. What are propionate and acetate doing?
They tend to act more systemically. Propionate and acetate circulate throughout the body, and they help regulate critical immune functions everywhere. And they also directly influence your overall metabolism, affecting things like glucose tolerance and fat storage. They're like direct chemical signals telling the rest of your body how well food processing is going down there.
It's not just energy and signaling, though. We're also completely reliant on them for certain vitamins, right?
That's a really crucial point of our coevolution. Microbes synthesize vitamin K, which is essential for blood clotting, and several B vitamins like folate and biotin that our own bodies cannot synthesize. So if you disrupt your microbial community significantly, you essentially disrupt your body's ability to manufacture these essential nutrients internally.
And they generate neurotransmitters.
Mhm.
This is usually the point that blows people's minds. The very chemicals critical for mood and cognition, serotonin, dopamine, GABA, are actually manufactured in high concentration in the gut, heavily influenced by bacterial activity.
Yeah, we often assume these are exclusively brain chemicals, right? But
Yeah.
the gut is, in fact, a major manufacturing hub for them. And this really sets the stage for our discussion on the gut-brain axis later, but it just underscores the fact that the gut is chemically communicative with the entire rest of the body.
Okay, so the final essential function we need to cover here is the defense barrier, what scientists call colonization resistance. This is perhaps the most practical, day-to-day function of having a diverse microbiome.
It's literally a battleground down there. Your beneficial bacteria compete fiercely with potential pathogens, the bad guys. They fight for nutrients. They physically block attachment sites on the gut wall so invaders can't latch on. And critically, they produce antimicrobial compounds, sort of natural antibiotics, and maintain the precise pH levels that harmful microbes just can't tolerate. It's a crowded, self-regulating ecosystem that ideally leaves no room for invaders.
And the real-world consequence of this defensive system failing is one of the most powerful illustrations of why this diversity matters so much. Can you walk us through the Clostridioides difficile, or C. diff, example?
Certainly. So when a patient takes broad-spectrum antibiotics, it really acts like a blunt instrument, as you said. It wipes out both the harmful bacteria causing whatever the original infection was, and vast swaths of the beneficial community that normally maintain that colonization resistance.
So the defensive line is just gone, obliterated.
Exactly. This disruption leaves the gut wide open, vulnerable to opportunistic spore-forming pathogens like C. difficile. Since the beneficial microbes are largely absent, C. diff faces no competition. It just takes over, proliferates rapidly, and starts producing toxins. And that leads to severe, sometimes life-threatening diarrhea and colitis. The failure of colonization resistance is what enables this condition. It shows just how critical a robust, diverse community is for simple internal protection.
That's a really powerful transition, actually, because if bacteria can influence our physical defense and our chemical production like neurotransmitters, it logically follows they might influence the brain itself.
They absolutely do. And this leads us to perhaps the most surprising, maybe paradigm-shifting discovery of the last decade or so, the profound influence of the microbiome on the brain, mood, and behavior. This connection established the critical concept of the gut-brain axis. We essentially have two major organs talking to each other constantly, and the microbes are actively participating, maybe even mediating that conversation.
So how are they communicating? Our sources detail, I think, 4 main pathways. It's not just one signal, it's a whole network, right?
Yeah, we can kind of break it down into chemical, electrical, and immunological communication. So first, revisiting our earlier point, neurotransmitter production. We now know that up to 90% of the body's serotonin, that crucial chemical for mood regulation, feelings of well-being, is produced in the gut. And bacterial activity directly dictates how much is synthesized and how it's released.
Okay, 90%, that's huge. The second pathway is through these microbial metabolites. We talk about SCFAs, butyrate, propionate, acetate. We now know these small molecules can actually cross the blood-brain barrier.
This is incredibly significant, yes. It means they aren't just sending signals locally to the gut wall. They are physically entering the brain. Once there, they can affect neuronal signaling, brain energy metabolism, and maybe even the structural integrity of the brain cells themselves. Direct chemical influence.
And the third mechanism is the physical superhighway connecting them, the vagus nerve.
Yeah, think of the vagus nerve as the central kind of two-way cable running between the digestive tract and the central nervous system. Bacteria in the gut can produce compounds, either metabolites or maybe bits of their cell walls, that stimulate the nerve endings embedded right there in the gut wall. This stimulation sends rapid electrical signals directly up to the brainstem. This is probably the fastest communication pathway, potentially informing the brain about the nutrient status or the inflammatory state of the gut almost in real time.
Wow, okay. And finally, the fourth mechanism is immune signaling. This connection sounds slower, maybe, but perhaps the most potent in the long run.
Exactly. Because the microbiome is constantly training and regulating the immune system, its influence can lead to the widespread production of cytokines. Those are those powerful immune signaling molecules. These cytokines travel through the bloodstream, and critically, some can cross into the brain. Once there, they can directly affect brain function, inducing what we sometimes call sickness behavior, feeling lethargic, withdrawn when you're ill, or in chronic low-grade forms, potentially contributing to psychiatric disorders.
The idea that chemicals produced by bacteria down in your gut can influence your mood, your behavior, it still seems almost like science fiction. But the evidence from animal studies here is incredibly compelling because it demonstrates causation, right? Not just correlation.
The evidence from germ-free mice is probably the most dramatic starting point. Scientists can raise mice entirely without any microbial exposure from birth. When you compare them to normal mice, these germ-free animals show profoundly altered brain development. They have abnormal and heightened stress responses and often display impaired social behavior. It clearly shows the microbiome is essential for wiring the brain correctly, especially in early life.
But the causation experiments are even more striking, those microbiome transfer studies you mentioned.
They are truly groundbreaking, yeah. For example, if you take the microbes from a mouse that shows anxious behaviors and transfer them into a calm, germ-free mouse, the recipient mouse actually begins to show anxiety-like behaviors itself. Conversely, you can transfer microbes from, say, a high-performance resilient mouse, and the recipient will demonstrate better coping mechanisms under stress.
And what about the human connection? Our sources noted transfer studies using microbes from humans with depression.
Yes, incredibly, studies have shown that transferring the microbiome from depressed human donors into germ-free rats can actually induce depression-like symptoms, like anhedonia, in those rats. This tells us the state of the gut truly can dictate the state of the mind to an frankly, alarming degree. And it raises the notion that certain behavioral traits or mood states might be chemically communicable via the microbiome.
Wait, let me just pause on that for a second. So we're essentially saying that differences in anxiety levels, stress resilience, maybe even depression could, at least in part, be driven by the specific mix of species inhabiting our colon.
Yes.
That's genuinely alarming, but also kind of paradigm shifting.
It absolutely is, because it gives us a completely new potential lever for intervention, doesn't it? In humans, this translates into striking correlations. Studies consistently find significant differences in the microbiome composition of people diagnosed with depression, anxiety, autism spectrum disorders, even schizophrenia when compared to healthy control groups.
Now, our sources emphasize that while the science is still, cautious about definitively stating that an altered microbiome causes depression in humans, the mechanisms linking the two are looking incredibly robust.
They really are. For instance, chronic inflammation, low-grade systemic inflammation, is now recognized as a significant factor in many psychiatric disorders. And that inflammation is often driven, or at least modulated, by the gut microbiome. A leaky gut barrier allows microbial products like LPS to enter circulation, triggering inflammation that ultimately affects the brain.
And the microbial influence extends right to the brain's own building blocks and growth factors.
It does, yeah. Through things like brain-derived neurotrophic factor, or BDNF. That's a protein essential for neuronal growth, plasticity, survival. The microbiome affects the levels of circulating BDNF. Furthermore, it directly influences the regulation of the body's stress response system, the HPA axis, and the subsequent production of stress hormones like cortisol. Your gut is constantly modulating how you perceive and react to stress.
So if the microbiome controls our stress response, influences chemicals essential for mood, it's clear we have to view the gut as a foundational pillar of mental health, not just a digestive tube.
Absolutely. The gut is, in many ways, the body's second brain, and its residents are actively shaping the signals it sends.
Okay, moving outward from the mind now. The microbiome is perhaps the most crucial immune trainer we possess. How does this partnership even begin? How does it work?
Well, the relationship between the immune system and our microbes is this incredibly intricate, ancient partnership. It's built on constant dialogue, constant interaction. And crucially, this training happens particularly intensely in early life, during that critical window when the immune system is developing its identity, learning what's self and what's other.
So early exposure to a diverse range of microbes serves as a kind of critical education for the immune system.
Yes, exactly. It teaches the developing immune system a vital skill: tolerance. It teaches the system to distinguish effectively between beneficial or harmless microbes, the friends, and truly dangerous pathogens, the foes. It helps the immune system understand what is self, what is foreign but harmless, like food or pollen, and what is foreign and dangerous.
And what happens if that essential education is missed, or maybe delayed, or incomplete?
The consequence can be significant immune dysfunction down the line. Without proper microbial education, the immune system can become hyperreactive, poorly regulated. It might start launching inappropriate attacks against harmless environmental substances, leading to allergies like hay fever or asthma. Or worse, it might start attacking the body's own tissues, resulting in the development of autoimmune diseases.
This sounds exactly like the basis of the hygiene hypothesis, doesn't it?
It is the core mechanism behind that theory, yes. The hygiene hypothesis proposes that the dramatically reduced microbial exposure we experience in modern sanitized environments, our obsessive cleanliness, smaller family sizes, less time outdoors, less contact with animals and soil, that this reduction directly contributes to the rapidly rising rates of allergies, asthma, and autoimmune diseases we see across developed countries.
Do we have strong evidence supporting this pattern?
We do, especially from large epidemiological studies. For example, studies looking at children raised on traditional farms who have high and diverse exposure to environmental microbes, animal dander, soil bacteria, consistently show significantly lower rates of asthma and allergies compared to children raised in highly sanitized urban environments.
And the connection is also visible in the effects of certain medical interventions, right?
Yes, particularly antibiotics. Early childhood antibiotic use, even just 1 or 2 courses, can profoundly disrupt this microbial education process during that critical window. Our sources confirm this disruption is reliably linked to an increased risk of developing allergic and autoimmune conditions later in life, basically because the immune system never received its full training curriculum from its old friends.
Okay, let's shift gears now to metabolism, because the microbiome's influence here extends well beyond just, what we typically think of as energy consumption. It seems to dictate how efficiently we actually use the food we take, in literally acting as a kind of secondary metabolic organ.
It really is an internal variation in energy processing. We've learned that different individuals extract different amounts of energy, different numbers of calories from the exact same meal. And this variation is heavily dependent on the specific efficiency of their unique microbiome composition. Some microbial species are simply far more adept at stripping those last few calories from complex carbohydrates than others are.
The obesity link, especially demonstrated through those animal models, is perhaps the most compelling evidence of this metabolic control, isn't it?
It's a very clear demonstration of causation, yes. Scientists took microbiomes from genetically obese mice and transferred them into lean, germ-free mice. The recipient mice, despite eating precisely identical diets in amounts to the control groups, gained significantly more body weight and body fat. This strongly suggests that the composition of the microbiome alone can drive metabolic change towards energy harvesting and storage.
So what are the mechanisms at play here? It surely can't just be simple calorie counting by the bacteria.
No, the mechanisms are incredibly complex and interconnected. We revisit SCFAs again as they affect fat storage patterns, glucose metabolism, and insulin sensitivity systemically. But the microbes also influence key appetite hormones directly. They can modulate the levels of leptin, the hormone that signals satiety, telling you when you're full, and ghrelin, the hormone that stimulates hunger, so they're potentially tweaking your appetite signals.
And we also see the link back to chronic inflammation here again. Correct. That seems to be a running theme.
Absolutely. The gut microbiome modulates the chronic, low-grade inflammation that is now understood as a key factor in the development of obesity and widespread insulin resistance. A less healthy, less diverse microbiome often leads to a more pro-inflammatory state systemically, which in turn makes metabolic control much harder for the body.
And then there is that truly intriguing speculation. I know it's harder to prove definitively that certain bacteria might actively influence our food cravings.
Yes, it's a fascinating idea. The concept is that certain dominant species within your gut might actually produce compounds, metabolites, or signaling molecules that influence your neurotransmitters or hormone levels in a way that creates a kind of feedback loop, pushing you towards consuming the specific nutrients, whether it's sugar or fat or fiber, that they need to thrive. So, are your cravings entirely your own conscious choice, or are they being subtly nudged by your internal residents? It adds another layer of complexity, doesn't it?
It really does. And finally, in this section, the microbiome has a direct and highly personalized link to cardiovascular health through what's known as the TMAO story. This seems like a perfect example of a dietary risk that is entirely dependent on your microbial inhabitants.
It is. This story centers on specific gut bacteria that possess the necessary enzymes to convert certain dietary compounds. These compounds are primarily choline and L-carnitine, which are found in high concentrations in foods like red meat, eggs, and some dairy products. When these bacteria encounter choline or carnitine, they convert them, producing a substance called trimethylamine or TMA.
And TMA itself isn't the main problem, but rather what our body does with it next.
Correct. The TMA gets absorbed into the bloodstream, travels to the liver, and there, liver enzymes convert it into trimethylamine N-oxide, or TMAO. And this final compound, TMAO, has been strongly and consistently linked in multiple studies to increased cardiovascular disease risk. It seems to promote atherosclerosis, the hardening of the arteries.
So this is why personalized dietary advice is so incredibly difficult. If I eat a steak and I happen to host a large population of those specific TMA-producing bacteria, my TMAO levels might shoot up, increasing my risk.
Yeah.
But if you eat the exact same steak and you lack those efficient converter bacteria, your TMAO levels might remain low and your risk profile is different.
That is precisely the mechanism. It's a stunning example of personalized risk assessment based purely on microbial composition. The food itself isn't inherently bad. The potential danger arises specifically from the interaction between that food and your particular gut community.
Okay, since we're clearly not born with this vast microbial empire fully formed, we have to acquire it somehow. And early life seems to be the critical window for colonization. The way we actually enter the world appears to dramatically affect our initial microbial seeding and really sets the stage for future health.
Yes, the mode of birth is absolutely a fundamental factor. Babies born vaginally are essentially coated, seeded, with the mother's vaginal and fecal microbes as they pass through the birth canal. This gives them a rapid, significant dose of the maternal gut community right from the start.
And C-section babies, by contrast.
C-section babies miss that initial seeding. They acquire a very different, often less diverse set of initial microbes. It primarily reflects the mother's skin bacteria and, importantly, the microbes present in the hospital environment, which can sometimes include less beneficial or even potentially problematic strains. And crucially, studies show these initial differences in the gut community between vaginal and C-section births can persist for months or even years, potentially influencing early immune and metabolic development trajectories.
And then there is the absolutely fascinating role of breastfeeding in actively sculpting the infant microbiome. It's not just nutrition.
No, breast milk is an incredibly complex, biologically targeted nutritional matrix. It contains these specialized sugars called human milk oligosaccharides, or HMOs. Now, here's the fascinating part. Human infants cannot actually digest these HMOs themselves. They serve no direct nutritional purpose for the baby.
So they're specifically there as food for the microbes,
Yeah.
like custom-made fertilizer.
Exactly. That's a perfect way to put it. HMOs act as specific, high-end, premium fertilizer, selectively feeding particular beneficial bacteria, especially species of bifidobacterium. This encourages their rapid establishment and dominance in the infant gut. And on top of that, breast milk also contains microbes themselves, viable bacteria from the mother, which actively colonize the infant's gut. It's biological engineering at its finest.
Amazing. So as the child grows and starts eating solid food, when does the microbiome start to look more like an adult's? When does it stabilize?
Well, once solid foods are introduced, the microbiome diversifies very rapidly. It's reacting to the sudden influx of different fibers, proteins, fats, varied nutrients. It typically stabilizes into a more adult-like profile somewhere around 2 or 3 years old, although the final composition continues to subtly shift and mature well into adolescence and early adulthood.
Looking at the really big picture here, this isn't some random arrangement that just started happening recently. This is clearly an ancient partnership, something driven by coevolution over hundreds of thousands, maybe millions of years.
Oh, absolutely. The human-microbe relationship is a fundamental biological agreement forged over vast evolutionary timescales. Humans evolved physiological features that actively support microbes, providing that stable, warm, oxygen-free, nutrient-rich habitat in the gut. And in return, the microbes provide essential functions that helped ensure our survival and success as a species.
We truly do rely entirely on them for certain things.
We do. Our sources really underscore that we rely on our microbial partners to digest the complex plant polysaccharides that likely formed the bulk of our ancestors' diets for millennia, and also to synthesize crucial vitamins we lost the ability to make ourselves. This reliance is now basically irreversible because we've shed the human genes required to perform these functions independently. It was an efficient evolutionary trade-off, you could say.
Yet this ancient, finely tuned partnership now faces a profound modern challenge. Researchers are increasingly concerned about what's being termed the disappearing microbiome.
Yes, These dramatic changes driven by industrialized lifestyles are highly sanitized environments, the widespread and often frequent use of antibiotics, especially broad-spectrum ones, and critically, a massive dietary shift toward processed foods that are very low in diverse raw fiber. These are fundamentally maladaptive changes for this ancient partnership. They disrupt the balance.
And the data actually supports this idea of lost diversity, right?
It does, yes. Studies comparing the microbial diversity of individuals in industrialized Western societies to those living in traditional non-industrialized communities like hunter-gatherer groups or rural agriculturalists show a really stark difference. Industrialized populations harbor significantly less diverse microbial communities overall. Certain bacterial species and even entire groups that were likely common or essential in our ancestors have become rare, or in some modern populations, virtually extinct. We've lost members of the team.
And this loss feeds directly into the old friends hypothesis, which seems truly crucial for understanding the current rise in so many inflammatory diseases.
The theory is very powerful, yes. It suggests that we evolved in constant close contact with a whole suite of old friends, not just bacteria, but also certain parasites, fungi, and other environmental exposures. These old friends were essential for training, challenging, and ultimately regulating our immune systems properly. By dramatically disrupting these ancient partnerships through modern hygiene, sanitation, C-sections, formula feeding, antibiotics, and diet, we've essentially removed the crucial braking mechanism on the immune system. And this dysregulation is thought to contribute directly to the current epidemic of inflammatory conditions like inflammatory bowel disease, IBD, allergies, asthma, and various autoimmune diseases.
This knowledge really begs the question. If the microbiome is so critical, what happens when it goes wrong, when it gets disrupted? And maybe more importantly, how do we begin to fix it?
Right.
When things go wrong, when the community balance shifts in a way associated with poor health, we generally use the term dysbiosis. Now, this refers to an altered microbiome composition, some kind of imbalance that is statistically associated with the disease state. But we absolutely have to stress here that defining a single healthy microbiome composition is incredibly difficult, maybe impossible, because the optimal state seems to be so highly personalized. What's healthy for you might not be optimal for me.
But regardless of that personalized ideal, we definitely know what the single biggest acute disruptor is, don't we? Without a doubt, the single biggest acute disruptor remains antibiotics. They are, of course, essential life-saving tools, we can't forget that, but they are inherently indiscriminate, blunt instruments. They kill both the good guys and the bad guys. Our sources clearly indicate that even a single course of broad-spectrum antibiotics can profoundly alter the microbiome's composition and function for months, or potentially even years afterwards. Some species might never fully recover, and repeated exposure, especially in early childhood during that critical developmental window, is powerfully linked to a higher risk of metabolic and allergic conditions down the line, likely due to this cumulative damage.
Okay, so if antibiotics are the acute disruptor, the sort of earthquake event, what's the chronic structural problem that erodes the ecosystem over time?
That would almost certainly be diet, particularly the modern Western diet. While antibiotics are like a one-time demolition event, the typical Western dietary pattern, high in processed foods, refined sugars, unhealthy fats, and critically low in diverse types of fiber, that's more like a chronic architectural problem. It gradually reduces overall diversity and actively promotes the growth of bacteria that are associated with inflammation and metabolic disease while starving out the beneficial fiber fermenters.
So the immediate fix, or at least the most fundamental approach to supporting a healthy microbiome, isn't necessarily super complicated from a biological standpoint, right?
Biologically, no. It's quite straightforward in principle. Diets rich in diverse plant fibers, think fruits, vegetables, whole grains, legumes, nuts, seeds. These are prebiotic foods. They actively promote the growth and activity of beneficial bacteria and encourage the production of those anti-inflammatory metabolites like butyrate we talked about. Basically, fiber feeds the star players on your team. Sugar and processed junk tend to feed the troublemakers.
This profound influence, both positive and negative, has obviously fueled the search for therapeutic manipulation. And this is where things start to get really complicated and sometimes controversial. The most common approach people hear about is probably probiotics, taking live microorganisms, usually bacteria, that are intended to confer some kind of health benefit.
Right. But the complexity here is just vast, and honestly, the results in many areas are still quite mixed. The effects of probiotics are known to be highly strain-specific. What that means is, One specific strain of a lactobacillus might show some benefit for a specific type of diarrhea, while another strain of the exact same species might have zero effect or even a different effect. You can't generalize. Plus, the evidence often suggests the effects are transient. The microbes don't actually colonize permanently, they just pass through. And frankly, the field is plagued by a lot of overhyped marketing that often outpaces solid clinical evidence.
So if the main issue with probiotics is that they might not stick around or colonize effectively, perhaps the clearer pathway seems to be prebiotics, feeding the residents you already have.
That is often considered a more reliable and perhaps foundational approach, yes. Prebiotics are essentially dietary compounds, primarily specific types of fibers like inulin, FOS, fructooligosaccharides, GOS, galactooligosaccharides. You're not trying to introduce a whole new team. You're simply providing high-quality fuel, specialized food, for the existing beneficial bacteria you already host, encouraging them to flourish. This approach generally has stronger evidence supporting it, particularly for enhancing beneficial metabolite production, like butyrate, and for improving certain metabolic markers.
Okay, then there is the really big gun of microbiome therapeutics. Fecal microbiota transplantation, or FMT. This is literally transferring the entire microbial community from a carefully screened, healthy donor into a recipient's gut.
Yes, and the success story of FMT in one specific critical area is absolutely phenomenal, treating recurrent C. difficile infection. For these patients, who are often desperately ill after multiple failed courses of antibiotics, FMT achieves cure rates exceeding 90%, often after just one treatment. It works by rapidly restoring that colonization resistance we discussed, essentially overwhelming the C. diff pathogen with a flood of healthy, diverse competitors. It's a biological knockout punch for that specific condition.
But if FMT is so incredibly effective for C. difficile, why are we seeing it used widely for other major conditions like IBD or obesity or maybe even autism, where microbiome links are suspected?
That's the crucial question, because applying FMT to these other, more complex chronic conditions has yielded really mixed, often confusing, and sometimes disappointing results so far. We simply don't yet understand the exact microbial signatures or functional deficiencies that need correcting in complex diseases like IBD or metabolic syndrome. It's not just about wiping out one bad bug. Furthermore, the risks associated with transferring an entire complex living ecosystem are substantial. Our sources clearly note the documented cases of recipients inadvertently acquiring undesirable traits from donors, things like gaining significant weight after receiving FMT from an overweight donor.
And the most serious risk, the one that really gives regulators pause.
The critical risk is the potential transfer of dangerous, unforeseen pathogens, particularly antibiotic-resistant bacteria. There have tragically been fatal cases linked to FMT where donors, despite screening, unknowingly carried highly resistant organisms that then caused severe infections in the recipients. This highlights the immense regulatory difficulty in classifying and managing FMT. Is it a drug? Is it a tissue transplant? How do you ensure safety with such a complex living therapy? The need for extreme caution and rigorous screening is paramount.
This difficulty in safely and effectively using the entire community really points toward the future, doesn't it? Towards next-generation therapeutics, moving away from the whole ecosystem approach towards much greater precision.
Exactly. The future is almost certainly targeted intervention. We're looking at things like defined microbial consortia. This means using specific, carefully selected combinations of cultured bacterial strains, maybe just 5 or 10 species known to perform a particular beneficial function, rather than the whole undefined community from a donor stool sample. Much more controlled.
We're also exploring postbiotics, which seems like perhaps the safest way to intervene of all.
Postbiotics is a really promising area, yes. This involves using the beneficial components produced by bacteria, or even structural parts of the bacteria themselves, for example, directly supplementing with butyrate or other SCFAs, or using specific cell wall components that have immune-modulating effects. You get the beneficial effect without needing the live bacteria at all. This completely removes the colonization hurdle and eliminates the risk of transferring live pathogens.
And then there are the really high-tech precision tools emerging on the horizon.
Right, those include things like engineered probiotics bacteria that are genetically modified in the lab to produce specific therapeutic compounds, like maybe insulin or anti-inflammatory molecules directly within the gut environment. Highly targeted drug delivery. And also phage therapy. Bacteriophages, or phages, are viruses that naturally infect and kill specific bacteria with incredible precision. We can potentially harness phages to selectively eliminate only the harmful bacterial species in a dysbiotic gut, leaving the beneficial ones untouched. This offers a level of precision far beyond what broad-spectrum antibiotics could ever achieve.
It's an incredible landscape of possibilities.
Yeah.
But despite all this progress, the field still faces some pretty significant scientific hurdles. The primary challenge, it seems, is constantly trying to distinguish causation versus correlation.
Yes, this is the researchers' biggest ongoing headache. When we observe an altered microbiome composition in someone with, say, autism or depression or IBD, is that microbial alteration the fundamental cause that initiated or drives the disease process? Or is it merely a consequence of the disease state itself, perhaps due to inflammation, altered diet, stress, or medications associated with the condition? Teasing apart cause and effect is extremely difficult in complex human diseases.
And establishing that causation often requires rigorous human experimentation, which is, as you said, difficult and ethically complicated to perform properly.
Exactly. And then the challenge of personalization complicates interventions even further. We know there's high functional redundancy in the microbiome. What that means is different bacterial species can often perform similar metabolic jobs. So even if you know a certain function is missing, targeting just one species might not fix it if others can compensate. Consequently, the same intervention, like a specific prebiotic fiber or a probiotic strain, will likely have wildly different effects on different people, depending on their unique starting microbiome. This necessitates highly personalized treatment approaches, and frankly, we don't yet have the diagnostic tools or the infrastructure to deliver that reliably on a large scale.
We also really need to look beyond just the bacteria, don't we? Most of the research, maybe understandably, has focused almost exclusively on the bacterial population.
That's a major, major oversight, and the field is starting to correct it. The overlooked members of the community are potentially equally important, maybe even more so in some contexts. We're talking about the virome, all the viruses, especially the bacteriophages that prey on bacteria, the microbiome, the community of fungi, and the archaea, another domain of single-celled life. The virome in particular may profoundly influence bacterial community dynamics by selectively killing off certain dominant species, thereby constantly reshaping the ecosystem and opening up niches for others to grow. We're just scratching the surface there.
And even when we do look just at the bacteria, the sheer volume of chemical signal they produce is overwhelming.
Oh, the metabolome complexity is a huge barrier, absolutely. We know microbes exert most of their important effects through the metabolites they produce, those SCFAs, neurotransmitter precursors, vitamins, etc. Yet there are literally thousands upon thousands of these small molecule compounds circulating in our body that originate from microbial activity. These are microbial signals affecting every single organ system. And the truth is, we currently understand the specific function of only a tiny fraction of them. We have the data identifying them, but we often lack the biological dictionary to interpret what most of them are actually doing.
Finally, before we wrap up, we need to touch on the ethical and commercial issues that are definitely starting to emerge as the science moves rapidly out of the lab and into the marketplace.
Yes, this is crucial. We've already touched on the regulatory struggles surrounding FMT. How should we classify this complex biological material? Is it a drug? A tissue transplant? A biologic? It's tricky, but perhaps more immediately relevant for many listeners is the risks surrounding commercialization, particularly direct-to-consumer microbiome testing kits. These kits often promise to unlock all your health secrets based on a stool sample, but frequently they provide results with extremely unclear clinical utility or actionable advice based on solid science. They risk overpromising health benefits that the current state of the science simply cannot reliably back up, potentially leading to unnecessary anxiety or costly and ineffective dietary restrictions based on flimsy correlations.
Not to mention privacy concerns, right? Your microbiome is pretty unique.
Exactly. Microbiome profiles are highly individual, like a fingerprint, and they can potentially reveal detailed information about your health status, your diet, your lifestyle, maybe even your susceptibility to certain diseases. This raises significant concerns about data ownership, privacy protection, and the potential for misuse of this deeply personal biological information.
Okay, let's try and bring all of this incredible complexity together. I think the key takeaway from this extensive exploration, this deep dive, is that we must fundamentally rethink our very identity. We have to start thinking of ourselves not as individuals, but as ecosystems. Our health is determined not just by our human cells, but by this complex, ancient, and constantly shifting interplay between human and microbial cells.
And that understanding is, I think, profoundly empowering, because unlike our underlying human genome, which is relatively fixed, the microbiome is highly modifiable through diet, lifestyle, and potentially therapeutic interventions. But it really requires a paradigm shift in thinking. It calls for a critical rethinking of many aspects of modern life, perhaps our over-reliance on certain antibiotics, our hypersanitation practices, our fiber-depleted diets in order to preserve and nurture the essential microbial exposures that actually helped shape our coevolution over millennia.
So here's something for you to mull over. If our microbial partners truly influence the food we crave, modulate how we respond to stress, and impact the foundational chemicals governing our mental health, to what extent are the decisions we make every single day truly entirely our own? And how might understanding and honoring our disappearing microbiome push us, perhaps necessarily, toward healthier, more resilient, maybe even more ancient ways of living?